So I'll start by re-acknowledging the group. Certainly, it's been an amazing journey with the groups that we have in this grant. So it's been a real pleasure. I'm actually going to be talking about just one Syntrichia genome. That's the Syntrichia canninervis. But we have also done a second one now, Syntrichia ruralis. So, but I won't be talking about that today. So the question became first, why would you want to sequence a genome? It's a lot of work and it's not exactly cheap. But if you're going to go anywhere to understand the biology and the biology of the moss, then certainly you need a background from which to do so. So the genome itself gives us lots of opportunities. One, first of all, it gives us the blueprint for constructing the moss itself. So you can start looking for parts of the genome that are involved in development or in any aspect of the biology of the moss. It's also a product of the evolutionary history of the moss, and you'll be learning more about that in the workshop as we go forward. So by looking at least having one standard genome that you've sequenced from an individual, then you can at least start to look at that evolutionary history of the moss by following that through different individuals and species. It's the genetic fingerprint for each individual. So each individual is going to be different, and that we take advantage of when we start looking at population genetics, which again, you'll be looking for later on. It also provides us all the genetic information that enables the moss to function in its environment. So that means, you can start looking at what are the genes that are involved in some of the basic functions. Certainly that's the physiology aspect of the grant. And when the environment changes, then obviously the moss has to react to that. And it does that by switching on and turning off genes or synthesizing proteins from a different gene set in order to respond to that environment. And the genome gives us the ability, along with the transcriptome, which is basically the transcripts that are derived from every gene, is what we call the transcriptome. By looking at that, you can start to understand what are the genetic requirements that allow you to survive that environment. And then finally, it gives us the genetic information that allows the moss to interact with its environment and with its neighbors, how it takes up nutrients, how it deals with competition, how it deals with other organisms that surround it. And those are, the chemical fingerprints or chemical stimuli that it will receive from the environment other than the normal abiotic types of events. And that's something, that allows, it gives us information on how the mosses survive in, say, a biological crust, which you'll obviously hear about later. So this is sort of how you take the basic genome sequences and start to look at that. So if you look at changes in gene and genome sequences between species over time, and now we have going on thousands of plant genomes now, and so only a few mosses, but that's getting more and more as we go day by day. You can start to understand the evolution of a particular individual, and in our case, the evolution of Syntrichia, where we can start to look at genes and how genes are organized in different species within the genus. Then differences in gene sequences between individuals. Obviously, that gives us a handle on population genetics, how those individuals react and form a community, a population. What are the genetics information that population holds rather than one individual holds. Then if you look at how different genes are expressed in different habits and environments, then you start to understand the genetics and the genomic aspects of ecology in the community. And that's an important area, our differences in how genes are expressed in response to changes in the environment, in the physiology. Then you start understanding how these bryophytes can have this sort of plasticity to survive and live in some very extreme environments. And then what's of specific interest to me, and certainly is a major part of the grant, is we've looked at how the genome setting and the changes in the genes that are involved in desiccation and rehydration responses in different species. And, you know, I've looked at that from level of the bryophyte as well as resurrection angiosperms. But it gives us now a handle on looking at how desiccation tolerance itself, which is a trait that is deep in the history of land plants. It gives us an idea of how that trait has evolved over time. So what did we discover? Well, this is the genome of Syntrichia cannonovis, and it's laid out in what we call a Circos plot. And it's designed at this point just to, around the different chromosomes, we found 13 chromosomes. And if you look at each layer of this, what looks like a big, to me, looks like a fruit flan, but that's being British, I guess, more than anything else. But you can see these different features of the genome, if you like. And the ones I want you to look at is the fact that the green connectors within the center of this pie, if you like, are areas of the genome that show collinearity. And so this starts to give us an idea of how different chromosomes are related to one another and how what's gone on in the evolution of this genome. But also it's interesting that we found one very large chromosome, and using some of Kirsten Fisher's markers, we could determine that chromosome 13, our largest chromosome, and the ones that actually has the fewest number of protein coding genes, if you like, is, we think that's our 6th chromosome, and the individual that we sequenced is a female. So this is really something that has fostered a few other inquiries and interest from other groups looking at the sex chromosome. We found that the genome is only about 300 and so megabases in size, so it's a fairly small genome, but it has 16,545 protein coding genes. And that's sufficient to do what it needs to do. So if we look at that idea of which different parts of the genome share collinearity with one another, it suddenly becomes, and we compare that to what has been posted as the 7 ancestral chromosomes. It comes out of a group in Germany. It's clear that the Syntrichia cannonovis genome has undergone one genome-wide duplication event. And there has been some chromosome loss. So chromosome 1, we only see one, if you like, copy of that. And that's true of chromosome 8. So we've lost 2 once we duplicated it. So you went from 7 times 2 to 14. We ended up losing 2. But the 6th chromosome, chromosome 13, has very little collinearity with any other parts of the genome. In fact, not even with chromosome 1 and 8 that were lost. And so we've got a gain of a chromosome, if you like, and that's how we end up with 13. So, and actually what's dropped off of this diagram is that we compared it also to Fischer-Matrella patterns, which is the best sequenced moss genome. Actually, it's called something else again now, Fiscometrium. But it also, it has a second duplication, so it's twice that. But the genome structure and gene contents also provide useful insights into different processes. So this is actually taken from a colleague of mine who works on angiosperms, desiccation tolerant angiosperms. These ELIPs, which are early light inducible proteins, protection proteins, they actually bind to the photosensors of the photosynthetic machinery and protect it from all sorts of things. Certainly, it's very important in the desiccation tolerance aspect of it. And what he found long ago was that the number of ELIPs genes that you find within a genome can be related to their ability to survive desiccation. And usually anything that's sensitive has less than 10 of these ELIPs, and anything that's desiccation tolerant has a lot more. And Syntrichia canenovus has 35 of them. So just looking at the genes and looking at for repeats and tandem repeats can often tell us how important genes are for particular processes. And then if you look at what genes are expressed at under certain conditions, and look at those sequences in a comparative way, if you like, you can start to see patterns of which genes are expressed under different conditions. So here we've looked at hydrated, dry, rehydrated moss that's in cold temperatures, and moss that's given a heat and a heat shock. And you can start to see patterns. And this one here, pattern 2, is actually shows a pattern that's related more to desiccation than any other of the patterns that we see. And so this tells us then, this gives us, if you like, a signature. And we can follow those signatures and look at how they are initiated in natural situations. And then we can say, what is this moss actually experiencing, and what is its major stressor? And if you can see, that's desiccation in this case. So, but these patterns also help us when we look at those in a genome context. And if you look now for genes that are in tandem, and then look at how they're related to their expression patterns, which are these purple and green layers, if you like, then you can actually pick out ones that are very important to desiccation tolerance, and they are actually in tandem repeats. So that means they're part of an adaptation, or they're more likely to be an adaptive change in the genome. And so we have this rubidoxin, which is an electron transport protein, which is very important in protecting photosystem 2. We have a Golgi protein, which seems to be important in protecting inner membranes of the cytoplasm. We have unknowns, which is always the case. So we don't know what that protein is or what those genes encode. And then we have a kinase, which is a very common kinase involved in signaling of water deficit responses. So overall, you can see how the genome now can inform you in all sorts of areas of biology, and certainly of the biology of the moss. And so this is sort of a summary, if you like. We know that we have the blueprint for the moss. It's made up of 13 chromosomes, 16,545 genes, sex chromosome. We know the genome was duplicated at some time during history. We know more about important genes that are amplified within the genome. We have signature genetic responses to environmental cues. And now we have novel genes that we can now start to investigate as their role in their ability to survive desiccation. So with that, I'll give you the main star of the show, if you like. This is Syntrichia canenovus in the Gobi Desert in China. And I threw some water on a patch there from my drinking cup. So, thank you.